For our final project, we decided to choose the Escherichia coli, specifically the O157H7 hemorrhagic colitis strain. So, the Escherichia coli is a commonly found bacterium in the human gut where most strains are harmless and even beneficial. However, this strain is highly pathogenic and can cause serious gastrointestinal illnesses. It is classified as an enterohemorrhagic E. coli and is primarily known as or known for producing Shiga toxins, which can lead to hemorrhagic colitis, which is bloody diarrhea, and in severe cases, the hemolytic uremic syndrome, commonly known as HUS, a life-threatening condition that damages the kidneys. This bacterium is important to study in multiple professional fields. For future healthcare providers, quick diagnosis and understanding of complications like HUS is critical. Veterinary professionals need to manage livestock, especially cattle, which are asymptomatic reservoirs for the specific strain. Public health officials and food safety specialists are responsible for preventing contamination outbreaks in food processing and distribution. Its relevance spans from hospital beds to food safety protocols, making this bacterium a key concern in both clinical and medicine to help for preventative health. So, E. coli O157H7 strain is a gram-negative rod-shaped bacterium. This means it has a thin peptoglycan layer sandwiched between two membranes, which causes it to appear pink when stained using the gram stains. The bacterium also possesses flagella, specifically the H7 antigen, which allows for motility and colonization. It uses fimbriae or pili to adhere tightly to intestinal epithelial cells, an essential step in establishing infection. Inside, it lacks organelles but contains a nucleoid region where its genetic material resides. These structural features support its survival in harsh environments and facilitates its ability to infect and persist within hosts. The outer membrane, known as the lipopolysaccharide layer, acts as an endotoxin and contributes to the inflammation and immune response. The Shiga toxins, though not physically part of the cell wall, are encoded by the bacteriophage genes inserted into the bacterial genome. These toxins are responsible for damaging blood vessels in the intestinal lining, leading to bloody diarrhea. The capsule can protect the bacterium from immune system defenses such as the phagocytosis. Additionally, the type 3 secretion system encoded on a virulence plasmid allows the bacteria to inject protein directly into the host to subvert normal cellular process. All of these components contribute to the high virulence of this specific strain of E. coli. So this E. coli strain reproduces through binary fission and can double every 20 minutes under ideal conditions. Like other bacteria, it follows a four-phase growth curve. In the lag phase, cells adjust to the environment and prepare for division. During the log phase, rapid and exponential growth occurs, and the bacteria are most metabolically active. This is also when Shiga toxins produce um, when Shigatoxin's production often begins. The stationary phase follows, where nutrient depletion and waste buildup causes growth to the level off as the rates of cell division equal cell death. Eventually, the death phase sets in as conditions become too harsh, leading to a population decline. Some cells may survive by entering a dormant state. As a facultative anaerobe, the E. coli O157H7 can grow in both oxygen-rich and oxygen-poor environments, helping it survive in food surfaces and the human gut. This adaptability contributes to its role in the foodborne illness and outbreaks. So, this specific strain of E. coli is a chemoheterotroph, meaning it must consume organic compounds for both carbon and energy. In lab settings, it grows well on general purpose media like tryptic soy agar or nutrient broth, but more specialized media is used for detection. On the conky sorbitol agar, this stain appears colorless because it cannot ferment sorbitol. Unlike most other E. coli strains that ferment it and turn the medium pink. This trait is crucial for identifying the O157H7 in diagnostic labs. It can ferment glucose and lactose, producing acid and gas as byproducts, which aids in basic differentiation from other enterobacteria. Nutritionally, this strain also requires nitrogen sources such as ammonium or amino acids for biosynthesis. It depends on trace minerals like iron, magnesium, potassium, and phosphorus, many of which are essential for enzyme functions and metabolic pathways. In the host environment, it uses siderophores to extract iron from host proteins, enhancing its survival and virulence. As facultative anaerobes, it can adapt its metabolism based on oxygen availability, switching between aerobic respiration and fermentation. And this metabolic flexibility allows it to thrive in diverse environments such as soil, water, and food surfaces, and the human intestine, making nutritional understanding essential for clinical and environmental control strategies. So this strand of E. coli is a facilitative anaerobe using aerobic respiration when oxygen is available and switching to fermentation in anaerobe conditions. It ferments glucose and lactose, producing acids and gases, but cannot ferment sorbitol, which is a key trait used in laboratory identification. This metabolic feature is what causes it to appear colorless on the McCongie agar to survive and grow in the host. This also produces cytophores, which help extract iron from the host tissue, supporting uh, both growth and virulence. Um, this strand contains a circular chromosome and plasmids that may carry genes for antibiotic resistance and virulence. Its most dangerous gene are the Shiga toxins genes acquired from the lysogenic bacteriophages. These toxins are responsible for causing hemorrhagic colitis and hemorrhagic uremic system or syndrome. This strain can also exchange genetic material through conjugation, transduction, and transformation, increasing its adaptability combined with mutations and more and mobile genetic elements. This genetic flexibility allows it to thrive in clinical and environmental settings.
E. coli 0157H7 is a dangerous foodborne pathogen that we aim to control primarily through food safety measure. Cooking meat to at least 71 degrees Celsius, especially ground beef, kills bacteria effectively. It is important to avoid cross-contamination in kitchens, keeping raw meat and vegetables separate, using clean utensils and surfaces. We also use cattle vaccinations to reduce the number of bacteria shed into the environment, decreasing the risk of meat contamination. Irrigation and pasteurization are industrial methods that help eliminate pathogens before products reach consumer. In addition, E. coli 0157H7 food safety protocols also target microbes like Salmonella, Listeria, monocytogenes, and Campylobacter, which can also cause serious illness as well. Detection of E. coli 0157H7 combines traditional microbiological techniques with advanced molecular tools. A common method is culturing stool samples on a sorbitol McConkie agar or SMAC agar, where E. coli 0157H7 forms colorless colonies due to its inability to ferment sorbitol. For faster, more specific detection, PCR testing is used to identify genes like STX1, STX2, and A, which with encode Shiga toxins and adherence factors. ELSA and lateral flow immunoassays can detect the presence of toxins directly. In large outbreaks, whole genome sequencing helps identify the strand and trace of the source. These techniques together provide a powerful diagnostic toolkit to confirm and manage infections. When E. coli 0157H7 enters the body, it first encounters our innate immune defenses. This includes the epithelial barrier of the intestines, which may become disrupted by bacterial attachment. The immune system responds with inflammation, recruiting neutrophils and releasing cytokines like IL-8 and TFN alpha, which is tumor necrosis factor alpha. The adaptive immune system then produces antibodies, specifically IgA, in the gut and IgG or IgM in the blood to target bacterial surface antigens and toxins. However, E. coli 0157H7 produces Shiga toxins that can evade immune defenses and enter the bloodstream. These toxins target the endothelial cells in organs like the kidneys, leading to serious complications. Unfortunately, the immune system often doesn't produce lasting immunity, and reinfection is possible. This causes a disease known as hemorrhagic colitis. This infection begins with severe abdominal cramps and watery diarrhea. Unlike many bacterial infections, it typically does not cause a high fever. The prognosis is good for most healthy adults, with full recovery in 5 to 10 days. A serious complication is hemolytic uremic syndrome, known as HUS, which occurs mostly in children and the elderly. HUS affects the kidneys and causes red blood cell destruction, low platelet counts, and acute kidney failure. It can be life-threatening. Treatment is supportive, focusing on hydration, electrolyte balance, and kidney monitoring. Antibiotics are not recommended because they may increase Shiga toxin release, which increases the risk of developing HUS. Antidiarrheal medications like loperamide should be avoided because they slow the gut and may prolong toxin exposure. If the patient's infection progresses to HUS, they may require hospitalization for close monitoring, blood transfusions to replace damaged red blood cells, and in severe cases, dialysis to support kidney function. With early medical intervention, most patients recover fully.